24 degrees centigrade. That is the vapor phase of liquid nitrogen. So we have 10 different cryo tanks in the cryo bank, and more than 820 different species they have been conserved. So these are some of the representative crops which are being maintained at the National Gene Bank at NBPGR. So through the NBPGR was established in 1976. Until now, we have conducted more than 3,000 explorations. And through these explorations and the exchange program with different countries, we have gathered around 4.62 lakh accessions, which are being conserved in the gene bank, and many more in the cryo bank and in the field gene banks. This is the different type of storage in the seed gene bank which we are maintaining. That is the short-term storage, medium-term storage, and long-term storage. So when we have to conserve the germplasm for a longer period of time, we have to process that germplasm in a very precise manner so that viability is not lost. Okay, so for short term, when we are conserving it for 12 to 18 months, we can keep the material in muslin cloth bags where air exchange can take place and there is no deterioration of the sample. In medium term, while we are conserving it for 10 to 15 years, the temperature has to be maintained and relative humidity has to be maintained. And then the samples are again kept in either in plastic bottles or glass bottles or even in paper bags. But when it comes to long-term conservation, when we are conserving the germplasm for posterity, that means for more than 100 years. So we have to be precise about the processing. So this processing is done in a very meticulous manner. As soon as we receive the sample, this is the flow chart, how we process the material at National Gene Bank. So we are conserving it in the gene bank, but if there is no information available there, what is the use of that? Okay, so we have to have the information about each and every accession. So whenever the material is received, we should have a complete passport data information about that sample, so that in future, if somebody else asks that I want this rice material collected from the you can say Coimbatore area or some villages from Coimbatore. So we can find it out from the passport data and send that same material to whosoever is requesting. So passport data information is very important. Then the material which we receive, since the crops are either they are self-pollinated or cross-pollinated, and our main motto is to conserve the whole diversity. So for self-pollinated crops, they are more homogeneous. So we conserve 2,000 seeds per crop sample. And for cross-pollinated, 4,000 seeds per sample. They are the mandatory seed quantity requirement. And then these samples, as soon as they are received, they are given a DQ number. DQ means domestic quarantine. When we receive the sample from outside the country, it is IQ, that is import permit or import quarantine. So all these samples, whether they are collected from within the country or outside the country, they have to pass through the quarantine division, because sometimes in some area, some virus or some bacteria is more prevalent than in the other areas. So to have a check on the domestic boundaries also, we are checking the material for DQ. So after this material, this has been passed through the quarantine division, it comes to conservation. Then we check the viability, because we are keeping the seed for 100 years. So it has to be have a very high viability. So more than 85% is the standard viability for any crop. But the relaxation is given in case of the wild species or difficult species, or in some of the vegetable crops where pollination is a problem. So we can conserve 1,000 seeds with 70% or even 60% viability, as is the case in the cotton crop. So once these samples, they have qualified all the different steps of the procedure, then they entered into the gene bank where they are given an IC number. IC is the indigenous collection. It is a unique number. Material which qualifies for the gene bank, it is given IC number, and which disqualifies, it goes back to the sender, whosoever has sent. And similarly, the material which comes from outside the country, that is given an EC number, that is the exotic collection. So this is the processing of the material. Like first cleaning is done, because if debris is there, there are more chances of having the deterioration of the seed sample. Then moisture testing, because for any seed to germinate, two conditions are important: temperature and moisture. If it finds adequate temperature and adequate moisture, the seed will germinate. So we have to restrict the germination by controlling these two parameters. So moisture content up to 5 to 7%, and uh, it is measured, and then the samples are tested for viability. If there is seed germination protocols available as per the E-star rules, we are following those E-star rules, but E-star rules are mainly for the crops which are across the globe. But for some of the indigenous crops, Indian crops, we don't have E-star standards. So we develop E-star standards in our labs, and then they are followed for the conservation. When the seeds physically they do not germinate, then we test the viability through TTC test. TTC is quick viability test. Okay. So there is a difference between germinability and viability. Viability includes germinated seeds as well as fresh ungerminated dormant seeds. Whereas germinability is only the seeds which physically show the presence of or the protuberance of the radicle. So after these tested, because the seed size and seeds uh, qualities are different, so we follow different methods, like top of the Patrick plate, or on between the paper, or in the sand also when the seeds are very bigger in size. After processing, the seed dried in a very um, well-designed, specially seed dryers, which maintain 15% RH, 15 degrees centigrade temperature, which allows this safe and slow drying of the samples. And once the samples they attain 5 to 7% in between the moisture content, they are taken out, and then they are uh, packed in a specially designed aluminum foul pouches, which are 3 layered. So outer layer is the polyester layer, middle layer aluminum foil, and inner layer is the polythene layer. And then they are vacuum sealed using the machine, so that all air is sucked out of the sample. Then conserving is one aspect. Maintaining the information or digitizing the information in computerized cells is a must, because unless and until you know the uh, material's identity, it is of no use for anyone. So all the information about any accession, that is the passport data information, which I will show you later on, that is fed into the computer systems, and we can retrieve the information based upon our query. So coming to the horticultural crops, the number of germplasm at the gene bank is more than 28,000. Genera represented 66. These are the species, and release varieties, around 1,000 release varieties of different crops that has been conserved in the National Gene Bank. So these are the cucurbitaceous crops, which are this map of India shows points from where this germplasm has been collected. Okay. And this is the diversity which is being conserved in the National Gene Bank in different cucurbitaceous crops. Similarly, in Abelmouscus, the diversity, species diversity, as well as the population diversity is conserved in the gene bank. And you can see the flower color and the size of the pod, different types, different colors, and even the taste is different. So all this diversity is being conserved in the National Gene Bank. In the solanaceous, you can see the brinjal, tomato, all these in the chili, the different diversity is being collected and conserved in the gene bank. So some of the underutilized vegetable crops, which are now becoming the potential crops for future, they are also being conserved in the gene bank, like Cyphomandra, which is a tree tomato, and then capsicum minimum, then Mamodica cochin sinensis. This is a very important crop. Nowadays, it is getting very popular in India as well as in the world because of its medicinal properties. Then Cannavelia, then Vicia faba, Sophocarpus. You see, these are some of the crops which are being now given emphasis for the as a potential crops in the gene bank. Similarly, in Coccinia grandis, then we have this Amorphophallus, Parkia roxburghii. Then these are the different uh, accessions or different collections for Mamodica. Then in Abelmoscus, very recently we have registered a germplasm with the ornamental okra, which is registered in the National Gene Bank. So this is the leafy vegetables. Again, amaranth, cholai, and fenugreek, coriander. And in case of medicinal and aromatic plants, we have around 1,000 accessions conserved and different genera which are being conserved. This is in ornamental genetic resources. Basically, all the ornamental crops, few of them have seeds propagated, but others are clonally propagated. So we have DFR in Pune, that is the Directorate of Floriculture Research, which is the national active germplasm site for floriculture or ornamental crops. So whatsoever material we receive, one part of that material is conserved in the field gene bank at DFR Pune. Then fruit genetic resources. India is very rich in indigenous fruits. So we have around 300 accessions of different fruit crops. This is the status of the gene bank. But in our field gene bank, we have more than 1,000 accessions of different crops which are being conserved. Then spices germplasm. Then another important activity at NBPGR is the trait specific registration of the germplasm, which I was telling you. So this mechanism was developed by ICR with NBPGR as the member secretary. And through this mechanism, you can register any germplasm which have any potential trait in your collections that can be registered through. Now this registration process is on online process, and you will get a certificate that this material has been registered with an IC number. And this registration is now gaining more popularity, and since its inception, 48 meetings have been held under the chairmanship of DDG Crop Sciences, and NBPGR, the head conservation, is the member secretary, and they conduct the meeting, and this germplasm is registered. So yesterday, the one student presented on cucumis. So he has identified one germplasm, and other student Ravi, he has presented a very early maturing Nigella germplasm. So this type of traits which can be of future importance for future breeding programs, this germplasm can be registered with ICR and BPGR. So for horticultural crops, we have total 440 germplasm which has been registered under these different crops. And you can see the photographs of some of these germplasm which has been registered. Like in case of vegetable crops, we have onion, then chili, which is a purple phenotype. Then this cucumber, which has high carotene content, and orange flesh. Then garden pea, and this one cucumber we have registered, which has two female flowers per node. Normally, there is only one flower per node, but this genotype has two flowers per node, and it is earliness and has a small fruit. Similarly, in case of medicinal and aromatic plants, we have this spiked ginger, uh, Hidicium spicatum. Then Malabar lemon grass has been registered. In case of isabgol, we have registered a germplasm which has golden yellow leaf color. Then Galenga, that is Campharia. And in case of Malexis, and then in opium poppy, opium poppy is a drug yielding plant, but this genotype, it is rich in the thebain content, and it has been registered by NBRI Lucknow. Similarly, in case of fruits, we have registered many of the fruit germplasm. Then this is in spices, different accessions which have been registered. In case of ornamental crops, all these flowering plants, they have been registered for different traits. So all this information is being digitized and is being compiled by our agricultural knowledge management unit. And these are the different PGR informatic applications available on our website. If anybody can want any information, they can go through these apps, whatever information they want, they can get. Then we have 6 databases, 10 applications, and 4 mobile apps like this gene bank app, if you download on your phone, you can have the information about the accessions conserved for a particular crop, or you can raise a query from a particular village of a state. All these type of different queries can be generated, and the information can be accessed from these different apps. So these are some of the um, photo shots of the different apps. Now it is the guidelines for sending germplasm. So if anyone wants to get the germplasm, either they want to send it for the germplasm for conservation in the gene bank, or for the registration or for the variety release, there are different processes and different information documents are required for that. Like for journal germplasm, we entertain only the mature clean seeds, which are free of any insect and pest. Then 2000 seeds for cross-pollinated and for self-pollinated, and 4000 for cross-pollinated. They should be packed in moisture permeable pouches, correctly labeled so that there is no misleading identity on any sample. Then seeds should not be treated with any chemical, because when we are conserving it for long term at -20, interacting with any chemical treatment on the seed, that might lead to any aberration or genetic disability in the sample. And then 5 different type of data which are required for any accession, which is meant for conservation, is the passport data, management data, characterization data, evaluation, and mode of reproduction. So what is the passport data? Passport data is all information about that sample. Okay, like you have your passport, and that passport gives exact information about you. Okay, your name, your father's name, mother's name, address, everything is there on the passport. Similarly, this passport data is for the sample. So it is necessary to give all the information, so that in future, if there is any loss from any habitat or from the gene bank because of some any calamity, passport data can help to relocate that sample, again go for collection, or to rehabilitate the same environment with the particular sample, if we have this information. Okay, so this information is important. Then the most important is the biological status in this information. Like what is the biological status of a sample? So biological status we have, after very detailed discussion, we have identified certain descriptors which give how you can identify the biological status for any sample which you are being collecting or you have been given access to. Like wild samples, all species other than cultivated species, they are conserved under the wild status. Then crop wild relative, which comes under the gene pool GP1 or GP2, and these can be called as CWRs. Then VD species are the VD forms of the cultivated species. Similarly, landrace, traditional varieties, farmers varieties, because there is a very little difference between a landrace and a traditional variety or a farmer's variety. So we have given certain particular descriptors which you can delineate between these two, between these three varieties. Then hybrids, inbred, segregating, breeding lines, potential genetic stock, registered germplasm, and elite lines. These are the different crop descriptors. Then release varieties, the release hybrids. Then there are state release varieties and institute release varieties. Some of the institute that CSIR institutes, they are releasing varieties on their institute level. Like we in ICR, we have a state release variety committee and then central release variety committee. But in CMAP or in CSIR institute, they have institute variety release committees. So NBRI releases their own variety, CMAP releases their own variety. So those varieties, they come under the institute varieties. Then exotic varieties, which comes through introduction. And if you don't know anything about that sample, about the biological status, you can give it others. So this was all about the conservation at ICR and BPGR. So if you have any query, you are most welcome.
Cultivar and release variety. And it is the cultivar and release variety like PK1 or Go1 like that.
Okay. Cultivar is any sample which is being cultivated. Okay, but variety, it should be released through a process or released through a mechanism. Like state release variety, it goes through the committee. Then there is a proceeding of that. Variety release proposal is there. So it will be a release variety. And cultivar is if you are not releasing it, but you are cultivating it, the same thing. That will be a cultivar, high yielding cultivar or sub improved cultivar, that you can say as a cultivar. Uh, yes. Okay. Oh, that is a release variety which is being released, like variety is released for a particular area. But yeah, that is cultivar. So it is a cultivar of that variety which is being grown in the particular area.
Morning, madam.
Yeah.
I'm Dr. Jagadishwari. So first of all, I should congratulate you, madam. You have given a very elaborative presentation. So students, they are all new students, PG students and PhD students. So we are dealing about the procedures, but we cannot know how the certification can be done. All those things you have given. The excellent presentation you have given, madam. Thank you. Then one more doubt for me. So I'm working in the plantation crops for a long period. So one major crop is coconut. So the coconut, it is a highly cross-pollinated crop and it cannot be multiplied through vegetative means.
Yes.
So we are maintaining active germplasm sites in so many places. So India is also one of the active germplasm center. So we have working with CPCRI.
Yes.
So now the trend is instead of importing the germplasm as the seeds, now they are going for embryo rescue technique.
Yes.
Yes, is it a viable technique or how long it can be stored, madam? How it can be preserved, the embryo, how it can be preserved?
So embryo can be conserved in the liquid nitrogen in cryo. So we are conserving the embryos of different crops now at NBPGR also, and then at the National Active Germplasm sites also. But for coconut, the development of that protocol is not till standardized. So we are under the process. One of my student has worked on the coconut also. So he has standardized some cryopreservation protocols, but the rescue of to the field is not as per the standards which we want. So that percentage is low. So wherever the standards are available, you can conserve the embryos in liquid nitrogen or embryonic parts also. We can conserve, like in Garcinia, the protocol is standard. So we can conserve the germplasm of Garcinia as a, you can say, safety duplicate, because maintaining trees in the field will be a more viable situation. But in case of cryoconservation, we can conserve the embryonic parts as a safety, you can say, safety duplicate also.
Now we have any number of embryos in CPCRI, madam? So that is the only center we have conservation of this embryo rescue. Whether you have it in.
Oh, it depends upon the number of material which we receive. Like if we receive adequate number of samples, we can conserve all. But in the safety duplicates, there are certain standards, number of vials and tubes per vial, how many embryos they are conserving. That standard is as per the cryoconservation protocol.
Thank you, madam. Thank you.
Good morning, madam.
Yeah, same procedure. For conserving tubers, we have different active germplasm sites which are conserving in field. But in cryopreservation, we are developing protocols for their clonally multiplication. So if the protocol is finalized, because these are all new crops which we are now taking up one by one. So when the protocol is identified, then they are conserved in the tissue culture facility, and then they are trained for the shorter duration, for conserving the germplasm for longer duration and maintaining them. So every time we have to regenerate, or we have to regrow those plants. So for that purpose, we have to identify the different protocols. So that process is still under taking place. So my colleagues at conservation division who are dealing with cryo unit and tissue culture unit, they will be more comfortable in giving these answers because I am mainly for the seed conservation.
Explained about so many methods, long term, short term, and medium term. So that is for the long term is for more than up to 15 years. Do you check the material in between for the viability? Or
Yeah, for long-term conservation,
Yeah.
because it is for posterity. Depending upon the type of crop, we monitor the germplasm. Like for traditional crops, after every 10 year or 15 years, wheat, rice, maize, this after every 10 year, we take out the sample, a small sample, and test the viability. If the viability goes down, then it is sent to the national active germplasm site for multiplication. Then again, the fresh sample is added to the gene bank. In few crops like oilseed crops, cotton, where the viability goes down very quickly.
So anybody attending through online, anybody having, if you have any doubts, you can please get it. You ask the doubts to the madam.
Yes, good morning. I'm R. Saraswati from Department of Plant Genetic Resources.
Your voice is not louder, madam. Your voice is very low, madam.
You can write down in the chat box that we can read.
I'm Saraswati R. from Department of Plant Genetic Resources. Is it audible? Is it audible? Am I audible, ma'am?
Your voice is very low, madam.
Very low.
Be louder. Or you can post your message in the chat box.
Ma'am, you can speak in loud, ma'am.
Hello.
Thank you, ma'am, for your valuable uh, lecture. We all enlightened our knowledge through your valuable words. Now, I would like to call Dr. Venkateshan, Head Department of Spices and Plantation Crops, on behalf of Dean HC&RI, Dean SPGS, uh, to give a memento to our chief guest, Dr. Veena Gupta ji. Now, I would like to call Dr. G.V. Rajalingam, Professor, Department of Spices and Plantation Crop, to deliver out of thanks.
Respected and our most distinguished chief guest of the day, Professor Neena Gupta, Madam, our beloved Professor and Head Dr. K. Venkatesan, sir, honorable professors, and my dear friends, I consider it a great honor to propose the vote of thanks to all who have helped us in making this seminar such a resounding success. First of all, I would like to propose hearty vote of thanks to our chief guest, Mrs. Professor Neena Gupta, Madam, for gracing today's seminar. Thank you, Madam, for your very interesting and thought-provoking address. I would like to thank our distinguished speaker, Mrs. Neena Gupta, Madam, for making excellent presentations and making this seminar interesting and meaningful. I would like to express our profound gratitude to our Dean, Ms. Dr. Iron Vedamani, Madam, for her help rendered in this seminar. We are also grateful to Dr. Sendil, Dean, School of Postgraduate Studies, for his help and support rendered in this lecture. I would also like to thank our beloved Professor and Head, Dr. K. Venkatesan, sir, for his moral support and guidance. I'm happy to express vote of thanks to Mrs. Dr. Mogan Lakshmi, Madam, PG Coordinator, and our staff members who have made this seminar a grand success. I would like to thank Ms. Vaishnavi for her active involvement in the seminar. And I also thank the PhD scholars, Abhinaya, Dibisha, Chandrakala, Akhil Raj, then MSc student Darshan Balaji and Mr. Nasir for their contribution. Finally, the wonderful students who have turned up in such great numbers, not only from our department, but also from other departments of the college. Thank you so much for your cooperation. I also thank the non-teaching staff who have helped in this seminar. Once again, I thank all for your cordial cooperation. Thank you. I thank especially Dr. Senthil sir, Dean SPGS, for sparing the hall for successfully conducting this event. Thank you, sir.
Thank you, sir, for your valuable vote of thanks. Finally, I hope all the participants enjoyed and strengthened their knowledge through this valuable program. Thank you, one and all participated here. Thank you.